Welcome to the Epigenetics Podcast from Active Motif. Join host Dr. Stefan Dillinger for lively discussions with leading epigenetics researchers. Hear about their past experiments, what they're working on now, and what's coming next. You know their papers, now get to know them and discover the stories behind the science.
Hello and welcome to this episode of the Epigenetics Podcast. Today I'm happy to welcome Weiwei Dang from Baylor College of Medicine on the show. Please let me briefly introduce you to our audience. You got your PhD from Southern Illinois University School of Medicine in 2006. You then moved on to do a postdoc with Shelley Berger at the Wistar Institute and the University of Pennsylvania. And then in 2013, you became assistant professor at Baylor College of Medicine. And since May 2023, you are associate professor there. A question I like to ask every guest to start off our little podcast is, how did you become interested in biology in the first place and then in pursuing a career in science?
When I was in high school, I was pretty good in math and science. I think more specifically physics. So I was doing very well. I have good grades, always do well in tests. Then I had a biology class, and I actually participated in one of those after-school clubs to work towards competitions with other schools, and I start to find biology fascinating. That's what really got me started on biology. Because before that, I was thinking I was just doing physics. I was interested in physics, interested in engineering, but then I started feeling physics so boring, and biology is really fascinating. There's so many different things, so many different possibilities. That's when I started becoming really interested in biology. That's the start.
Yeah. So when you started in college, or let's say your academic career, did you then already know what it would mean to start an academic career? Because I didn't know when I started at the university, I didn't know what it meant to do a PhD, but did you know what you signed up for?
Not really. It really... start understanding it when I started my PhD study.
So when it was already too late to go back.
That's already too late. Yes. Well, in fact, a lot of my friends and classmates in college went the same way as me. I guess it was like the trendy thing at that time. A lot of Chinese students in these top schools, top colleges, seek to pursue like PhD in the U.S. in science. In fact, I would say 70 to 80% of our class went to the U.S. After graduation. So that's the kind of thing everybody was doing, and so I was doing it. And then it turns out many of them eventually changed their career, changed their subject. Many of them finished their PhD, but eventually changed the subject of study or the field of study, field of working, I guess. Got a science degree, but worked in other industries.
Okay.
And some of them even changed their degree of study or subject of study. They quit PhD program and they apply for another program. I know a friend who studied finance later on. I have another friend who studied law. So there, it becomes very diverse in the end.
Yeah. But let's talk about your science that centers around the molecular mechanisms of aging and how to delay aging and the onset of age-related diseases such as maybe cancer. I think it was when you were working with Shelley Berger, when you're interested in epigenetics and aging started. In her lab, you looked at two histone PTMs, namely H3K16 acetylation and H3K36 methylation and their connection to aging. What did you find about the connection of those histone marks to aging, and was there even a causal link?
Yeah. So when I joined Shelley's lab in 2006, I was given two projects, or basically two questions. One is more traditional, looking at another PTM mark. There was a paper published just a few months ago on another novel PTM. I was asked to see if that PTM also happened in yeast. It was first found in, I think, frogs. And so that was more like a traditional type of project for the lab. Just found a new PTM to study that PTM in the system that we're used to work with. Well, another project or sort of question is aging. At that time, Shelley's lab started to move into this general area of how histone PTMs are involved in various physiological processes. In the past, the field focused on how it regulates transcription, DNA repair, DNA replication, all those basic molecular processes. But then we started looking at various physiological functions. So that's the question, is to look at the PTM's role in aging, histone PTM's role in aging. No one else in the lab worked on aging. And so as a start, of course, first of all, I read literature. I happen to have a colleague at Penn, another professor who at that time was in the aging field for a long time. He was trained with Lenny Grante. He's one of Lenny Grante's postdoc. He was also studying yeast. He's studying yeast telomeres. I talked with him. He was like my mentor in the aging field, so to speak. And then I attended a meeting on aging. The first meeting I attended was Cold Spring Harbor aging meeting. That happens every other year in the fall. That really got me interested in the aging field in general. Aging is one of those few field of biology that almost it touches on every other part of biology, and it's again rapidly growing, and there's so many unknowns in there, so many things that you can do. It's really fascinating. Well, another thing I noticed in that first meeting was almost nobody was looking into the chromatin or histone modifications, or what histones are playing a role during the aging process. At that time, most people focus on metabolism. That certainly makes sense, because we know dietary restriction extends lifespan. It certainly have tremendous effect on metabolism. And there's also a lot of interest in various stresses, like oxidative stress, mitochondria, all that stuff. Nobody was looking into chromatin. Even though one of the enzymes that are known, the famous enzymes that are known to regulate aging, the sirtuins, are known to regulate histone PTMs, but nobody was really looking into chromatin histone's role during aging. And so I thought that was my opportunity. And so that's what really motivated me to pursue this project.
Mm-hmm. And what did you find out about the function of those two histone PTMs on aging?
Basically, our system is yeast. So I learned about how yeast can age. I didn't know about that. So in fact, there's 3 different ways to look at yeast aging, surprisingly. So the model that we chose to use is called replicative aging. So basically, the budding yeast cells produce daughter cells by budding. So by budding, you can imagine that it forms a small cells out of a big cell, basically. And so it is asymmetric, and you can distinguish the daughter from the mother. And so you can track and monitor the mother cell's division and see how many daughter cells it produces. And so that's basically called its replicative lifespan. On average, the wild-type cells produce about 20 to 25 daughter cells. So this is the one of those model systems that really got the aging field started. Letty Grante was one of the pioneers in the field using the budding yeast and discovered sirtuins. And the other model being C. elegans. That's work from Cynthia Kenyon and Gary Rufkins, who worked on C. elegans, discovered various pathways in C. elegans. Well, so back to yeast. So we learned a lot of how to analyze aging in yeast from Lenny and David Sinclair's work. So I started using those methods. One of those methods I learned from Lenny was to isolate aged yeast cells by something called surface labeling. And that can be done because when yeast produce the bud, the daughter cells, the entire cell wall of the bud is newly synthesized. So if you label the mother cell's cell wall, that label never goes to the daughter. It sticks with mother. That way, you can age a bunch of cells. And so you first label a small population of cells, then age them, and later on you can retrieve these labeled cells when they're old by affinity purification. So now we can isolate these old cells in large quantity. Of course, the young cells are just abundant. We just compare their PTMs. We used all the enzymes available in the lab to test which of histone modification changes. And it turns out the first thing we found was H4 lysine 16 acetylation. It significantly increases in the old cell population. So that was the first indication that's and sort of answer the first part of the question is whether or not PTM changed during aging. So we found the answer to the first part of the question, and then we wanted to ask whether or not that change is causal, whether or not it actually play a role in regulating aging or causing aging. And so that to answer that question, we took advantage of the yeast model. It's because manipulating histones are really difficult in other mammalian systems, other eukaryotic systems. But in yeast, it's relatively easy. It's because yeast has only 2 copies of each histone gene. the major histone gene in the genome, in the haploid genome. That compares to like over 200 copies in flies, more than a dozen in the C. elegans and mammalian systems. So in terms of the ability to manipulate histones, yeast is a wonderful system. So people already have done that in the yeast, in the field, is to mutate histones, histone residues. So we could mutate H4K16 and see if that changes the lifespan. So that was one of the follow-up experiments I did. I mutated the lysine 16 to arginine to glutamine. Mutating it to glutamine mimics the acetylated state. And so that's sort of well accepted in the field. So I made the mutation. Mutating to arginine mimics unmodified state. Mutating to glutamine mimics the acetylated state. So those are the two mutations I made at the beginning. And then first I made it in the, it's a little more complex. First I made it in the plasmid. That was the standard way of operation at the time. And very quickly, we got the result, making it, mutating it to lysine to glutamine to mimic a acetylated state really accelerates the aging process. It makes it lifespan much shorter. And so that sort of proved that it's a causal relationship. Mm-hmm. And on the other hand, we can also look at the enzymes that regulate this mark. And especially in yeast, in the yeast system, the enzymes that regulate these PTMs are very clear and very simple. There's usually one enzyme that puts the mark on and one enzyme that takes it off. In this case, H4K16, there are two different acetyltransferase that make this mark, but one enzyme that actually focuses on the subtelomer region. So that's really convenient. And then there's the sirtuins that work at on removing the mark, right? So we looked at these two enzymes mutations. Sirtu, as everybody know, but at that time shortens lifespan, so it matches perfectly with our observation of the K16 mutant. So we made SAS2, which is the acetyltransferase mutant. SAS2 mutation actually extended the lifespan. So you have less of the acetylation, the subtelomere region extended the lifespan. So that really is the hard evidence to support that it is a causal effect. Mm-hmm. When you have more of it, it shortens lifespan. When you have less of it, it extends lifespan. So you mentioned another mark, H3K lysine 36 methylation. That's another mark that we worked on. I started working on towards the end of my postdoc with Shelley and continued on in my own lab. That's another example of it. Mm-hmm. The case 36 methylation, the enzymes has these opposite lifespan effect. You mutate the methyltransferase, which is SET2. It shortens lifespan, and then you mutate the demethylase, extended the lifespan. So those are examples of mm-hmm. those causal evidence for aging.
So when you transitioned from Shelley's lab to your own lab, so you took basically this aging or this interest to the aging process with you, right? And you worked on it then in your own lab.
Yeah. So over the year, I basically built up on my interest over the years. So remember, I started with 2 projects, and the other project didn't go anywhere. And then I eventually focused on aging. And after the first story was published on the H4K16, I really expanded my aging projects, the questions that we're asking.
Mm-hmm.
I started doing more screen or high throughput type of experiments. I want to find all the histone residues are regulating the aging process. I want to find all the mutants in yeast that I can that regulates aging. So I started working on high throughput screens with a focus on chromatin regulators. The first such screen was a histone mutant lifespan screen. So that's another example of how powerful yeast genetics is, where it's so hard to manipulate histone in other systems. In yeast, we actually mutated every single amino acid histones, and there are 2 different libraries available. So we used one of those libraries and designed a high throughput screen. The traditional lifespan screen in yeast was done by manual dissection, and it's very tedious, very time consuming. So I designed this O-cell purification based high throughput screen method. Basically, we start with all the mutants together and age them, and then purify all these old cells, like using the method I described early on. And then you can imagine the short-lived ones will drop out and long-lived ones will stay. And all we need to do then is to figure out which one dropped out, which one's enriched. And we can do that by barcode sequencing, because all the mutants in the yeast mutant libraries are barcoded on the DNA. So that makes it very easy to identify which mutant they are. So we did barcode sequencing to figure out which mutant dropped out, which mutant got enriched. So we started to do screen like this, and this actually led to a lot of future projects in my own lab. So this is the basis of my own lab projects. Is
the first screen.
The discoveries in yeast.
Yeah, what did you find doing those screens? Was it more finding new histone modifications, or were you more successful on the complex side of things?
Yeah, so one of those things we found was the H3K36 site that you mentioned early on. We found that through the screen, we found that K36 mutations, almost all the mutations that exist in the library, shortened the lifespan. So then we decided to, and because this site is known to be methylated and it is known to play, because this site is methylated, and we look at the methylation enzyme. So we basically put all the evidence together, and this is one of those sites that had consistent effect. That is, the mutations are short-lived. The enzyme mutations all match up with the site mutations. So that's why we decided to follow up on this site. And because this site is also, site's function is also kind of known in the field. It's due to the work from Jerry Workman in the field, who discovered that H3K36 methylation helped to suppress something called cryptic transcription. Cryptic transcription can be seen as the aberrant transcription that initiates from inside the gene, not at the promoter. And it happens when you have mutants that have either defects in transcription elongation or have a defect in H3K36. When you lose this mark, everywhere in the gene is in the open configuration, because normally when a gene is transcribed, it assembles the initiation complex in the promoter. As soon as that's assembled and ready to go, it transitions into an elongation form, and then to finish the transcription process and produce the mRNA. And so while it's doing the transcription elongation, it recruits the SED2 or SED2 in the mammalian system to make the K36 methylation along the gene body. Okay, so for actively transcribed genes, the promoter and the gene body actually have different histone modifications. The promoter is decorated with K4 trimethylation, the gene body has K36 trimethylation. It's very different environment. K36 methylation in turn recruits other enzymes to help actually close down the chromatin, to maintain a relatively closed conformation in the chromatin for the middle of the gene. And yet that leaves the promoter in an open conformation. It allows transcription to initiate again, next round of transcription. Well, when you mutate this SED2, you lose the H3K36 methylation altogether. You have none. And then what happens is in the gene body now is open. It cannot be closed after the round of transcription. So the entire gene looks open from the promoter to the end of the gene. Any sequence that look like promoter basically is rich in TATA sequence, can initiate transcription. So that's how these cryptic transcripts are generated from promoter-like sequences, or they call cryptic promoters. It initiate transcription, usually in the gene body, and proceed to the end of the gene. So generating these short transcripts. So these can be seen as a transcription noise, or so this is a great model, model, right? It's very clean, very clear, and this model now we know that is conserved even in the mammalian cell. But back then, it's considered just it was only worked out in yeast. But even in yeast, it was very puzzling at that time, because many of these mutants have no defect, no observable defect, especially the SED2 mutant, the mutant that lacked the K36 methylation at all. It had very little growth defect. So it puzzled the field for a long time. What is the benefit of suppressing cryptic transcription? Until we found that it's probably playing a role during the aging process, right? Because we found that these mutants actually have an aging phenotype. So we started to question, maybe in old cells we do have more cryptic transcription. And so it was, at that time I had a different project. The other project was to look at the proteome. Like I said, after my first paper came out, we started looking at many different questions. So one of the other questions was to look at how proteome has changed during the aging process, not just looking at histone, but every other protein. So we collaborated with another lab to do quantitative proteomics. But at the time, I was thinking, we do proteome, we got to look at transcriptome as well to understand how proteome is changing. So I started to collaborate with a sequencing lab to do the next generation sequencing, RNA-seq. And we just happened to have that data available. We just got the RNA-seq data. And together with the screen and the lifespan data, so we predict that there are more we predicted that K36 because the K36 mutants and the gene mutants, the related enzyme mutation have the effect. We predict that there are more cryptic transcription in the old cells. We got into the RNA-seq data set. We look at the genes. We basically pile up all the RNA-seq reads on genes and look at the patterns from young to old. And all of a sudden, we saw there's increased RNA signal towards the 3' end of genes. And it happens in many, many genes. We started categorizing this effect. And we, even if you pile up all the genes together, you see increase in 3' end signals. But then as we are doing different categorization of these, we've realized it happens more often in the long genes. And that makes perfect sense, because the longer the gene is, there's more chance you have a promoter-like sequence embedded in it, and there's more chance we have cryptic transcription. Okay, so that's how we started this project.
So
and then
did you find any effects of this cryptic transcription? I mean, there is then a lot of RNA piling up. Maybe this RNA gets made, or does it get made into proteins? Or what is the negative effects?
Some of them do get made into proteins, and the truncated proteins have been detected in yeast. But to this day, the downstream effect is still not clear. But at that time, what we can show is that these cryptic transcriptions are produced during in the aged cells, and then if you suppress them by improving the K36 methylation levels through deletion of the demethylase, it can suppress the cryptic transcription, and it can extend the lifespan. That's the key message we had with this paper. That is, this is another of those examples where we found a mechanism, a chromatin mechanism that causatively regulate the aging process.
You just went over my question, or another question that I prepared in one sentence, because you said it's shown in mammals as well. You tested this, right? So
yes.
To what extent is this also present in mammals? And can you compare it to yeast and other maybe life forms?
Yes. So naturally, our ultimate goal is to understand human aging and what happened to our cells during the aging process. So we found this mechanism in yeast. Naturally, we are looking for evidence whether or not it's happening in other eukaryotic system. We usually first look into C. elegans or flies. So we did that for this project as well. We looked at C. elegans first. There's cryptic transcription. A homologous enzyme knockdown also had a similar effect in extending lifespan. So with that evidence, we started looking in mammalian cells. We started with the RNA-seq data set in the senescent cells. We found evidence for that, for cryptic transcription. And then we started looking into published data sets using mammalian stem cells, the mouse hematopoietic stem cells. That beautiful data set was produced by Peggy Goodell's lab at Baylor. So this is the work I started when I had my own lab at Baylor. So we started looking at this mouse hematopoietic stem cell RNA-seq data set. Those hematopoietic stem cells were isolated from young and old mice. And we found cryptic transcription through the RNA-seq. Of course, there's some technical details I don't have time to get into, but we could show that we have evidence that there are cryptic transcription there. And in my own lab, we started using a stem cell culture system. We get stem cells from umbilical cord tissue, those so-called mesenchymal stem cells or mesenchymal stromal cells. These cells can be differentiated into different lineages. They are primary cells, so they can be serial passage, but they have they're regulated by senescence. Eventually, they reach senescence; they cannot proliferate anymore. Sort of like the aging process, but happening in vitro in petri dish. So we started looking at so these cells before they reach senescence, you can already observe functional change in that they gradually lose their ability to be differentiated into different lineages. So we thought that that's our entry point. We can look at this aging process, this in vitro aging process, and look for what roles the chromatin is playing, various histone modification and chromatin state is playing in this process. And so one of those first things we looked at, we did with RNA-seq, and then we looked for cryptic transcription in those cells as well. So these are the two starting. We found cryptic transcription in both cases, and then we started downloading and collecting all the RNA-seq data sets that we can find from the database, whether it's either aging or senescence in mouse tissues or human tissues or cells. Of all the data sets we examined, I would say more than 90% of them we can detect cryptic transcription associated with either aging or senescence. And so this is just a relatively simple analysis from RNA-seq data set. Of course, we want to understand what's causing these cryptic transcription, and we wanted to understand the chromatin's role in causing these cryptic transcriptions in aging or senescent cells. So we used our mesenchymal stem cell model to do a lot of ChIP-seq experiments to look at the chromatin change. We used a novel RNA-seq experiment called DCAP-seq that specifically captured the 5' end of the mRNA to help us understand where the promoter is actually located and pinpoint the location of the cryptic promoter and quantify the cryptic initiation compared to the regular promoter. So these data help us understand what's going on at the cryptic promoter, what happened to the chromatin at the cryptic promoter that allowed them to be activated. And the conclusion from this study, this analysis, is that the chromatin at these cryptic promoters, or these promoter-like sequences that's embedded inside the genes, become more open, more permissive. That's the word we used. Become more permissive. Because these sites are really promoter-like sequences in that they're recognized by TBP even in young cells. We found TBP binding to these cryptic promoters or promoter-like sequences inside the genes even in young cells. But because the chromatin is closed, it's repressive, because of the role of H3K36 and its associated DNA methylation in mammalian cells, chromatin is closed in young cells. But with age, this repressive chromatin environment in actively transcribed genes in the gene body become compromised, becomes more open, more permissive. It allows the assembly of the transcription initiation complex at the TBP binding sites, then cryptic transcription occurs. Mm-hmm. So that's the story that we published a couple of years ago.
Then what are you working on right now and maybe for the next 5 years?
I can tell you a really interesting project or direction that we're working on. That is to help define another phenomena that another mechanism we discovered in yeast, and we're actively working on this phenomenon in the mammalian system. This is something we called chromatin stress response, or the official term that we coined was chromatin architectural defect response. Mm-hmm. This was discovered through our studies of yeast mutants that yeast the yeast cells with altered histone dosage or altered histone copy number. Remember, I told you early on there are 2 copies of each histone genes in yeast.
Yes.
You can knock out 1 copy and leave just 1 copy. The yeast is fine, it grows well. We can titrate the histone copy number, change the histone copy number, essentially titrate the histone dosage. What's also interesting is that the 2 copies that's in yeast, although they produce identical histone proteins, these 2 genes coding sequence are different, and their relative expression levels are different. Okay, so they have about a 1-to-6 ratio. Mm-hmm. So this is what we started. We knock out 1 copy. We call them minor copy. We knock out the minor copy. You reduce the histone dosage just by a little bit. Or you can knock out the major copy. You reduce the histone dosage by quite a bit, a lot. Well, we compare the lifespan. Well, you reduce by a little bit, it actually extended the lifespan. Okay, if you knock out the major copy, you reduce quite a bit, it shortened lifespan. That's sort of expected. When you have a lot less histones, cells are not able to live long. And in the past, we also showed that in the old cells, histone protein levels are reducing. Okay, so the simple prediction from that is when you have less histones, you have shorter lifespan. When you have more histones, you have longer lifespan. And Jessica Tyler's lab actually showed when you overexpress histones, extended lifespan. So is this just a simple correlation? And we showed in our histone dosage experiment, when you reduce just by a little bit, it actually extends the lifespan. It extends lifespan even more robustly and consistently than if you overexpress histones. So this got us interested to try to understand why when you reduce a little bit, it has an extension effect, lifespan extension effect. When you reduce quite a bit, it shortens lifespan. How can you explain? We started out with RNA-seq experiment, try to understand what's going on in these cells. They turned out to have very similar RNA-seq profiles. It's very surprising, even though they have very different lifespan results. And so then we realize there's something similar between the two, and also between the histone mutants with other gene knockout mutants, certain other gene knockout mutants. And we realize that they're very similar with disruptions in the chromatin assembly mechanisms, pathways. They have very similar transcriptomic profiles with histone chaperone mutants. Okay, so these mutants share similar transcriptomic profile. They also share similar metabolic phenotype. They have increased respiration, increased mitochondrial copy number, increased ATP production. So they have similar transcriptomic profile. They have a similar metabolic profile. And that's why we started thinking, oh, maybe this is a new form of stress response. There is stress response that's activated by defects in chromatin. So we named it chromatin architectural defect response. And then we later on we found that there's a set of genes that are upregulated under this condition, and then when you reduce the histone dosage, and these genes are regulated by a common transcription factor called GIS1. And if we knock out the GIS1 transcription factor, you don't have this transcriptional effect, then cells live shorter. And if we overexpress this GIS1 alone, can extend the lifespan. So it basically prepares the cells to respond to stress later on in life better, because histone dosage reduces, and the cell needs a chromatin stress response to compensate for the loss of histones. And this mechanism is there to help. So again, this is a brand new mechanism that we discovered in yeast, and we wanted to see if it's happening in mammalian cells. We have some evidence based on some data analysis, but we're now actively trying to prove that this is happening in the mammalian cells, and we wanted to investigate whether or not this kind of response can, what kind of a physiological effect it can have in the mammalian cells when this is activated. So this is one of the area my lab is currently focusing on. Mm-hmm. And something related to this is about cancer development, right? A few years ago, in the past, people know that the chromatin and the histones are regulating aging, regulating cancer development. But it really become striking to that histone mutations were discovered to be discovered in various human cancers, and eventually the histone mutations are proved to be drivers of oncogenesis. And it's shown that some of the histone mutations can cause massive change in histone modifications, but other histone mutations, rather than cause changes in histone modification, they disrupt nucleosome stability. They cause destabilization of nucleosomes. So this got us to think that if these cancer cells have destabilized nucleosomes, you got to activate its chromatin stress response that we discovered. Now, what does this mechanism play a role during oncogenesis? Well, we know a couple of other stress response pathways promote cancer development. Things like hypoxic response, things like oxidative stress response. They help the cancer cells live through these stressful conditions and become eventual, nonstop proliferation. And maybe this chromatin stress response pathway also playing a similar role in that it support these cells live through conditions of chromatin stress, because this mechanism help promote cells proliferation, cell survival, and longevity. It helps the cancer cell to become cancer cell.
Mm-hmm.
So this is a novel direction that we're working on to understand whether or not this mechanism has a role in cancer development.
Okay. So in the last 53 minutes, we have been on a journey through your scientific career. Did we miss something important along the way, or would you like to add something?
I think we talked. I think we discussed all the major aspects of my career now. Yeah. I want to add that my PhD training was on not on histone modification, but rather on ATP dependent chromatin remodeling. It's a non-covalent change to the chromatin by again a set of enzyme that's highly conserved from yeast to humans, and they're called ATP dependent chromatin remodeling enzyme. So my PhD project was to work on one of these enzymes, how they interact with histones. It's very biochemical, pure biochemistry, in vitro study. I remember at that time, my mentor was getting grants, either getting grants or getting paper published. The criticism was, it's all in vitro. It's not physiologically relevant. So that's why I wanted to switch to a system where we actually study in vivo phenomena. Well, so that project helped me to start another project that later on we studied a chromatin remodeling enzyme and its role in regulating yeast aging, and that is called ICY2. It's the same enzyme I studied during my graduate school. Mm-hmm. It turns out this enzyme also regulates aging, and it regulates aging through its role in regulate stress response gene expression. So that's a really interesting coincidence. I get to work on the same protein I work on during my graduate study, and published a story that the same thing regulate the aging process.
So you're working on the same thing, but just in a different context.
Very different context, yes.
So I guess then this is a good time to end this interview. Thank you, Weiwei, for your time and for being on the show.
Thank you very much for the opportunity. Really nice talking to you.
Thanks for listening to this episode of the Epigenetics Podcast from Active Motif. We hope you enjoyed it. You can find all the mentioned references in the show notes. Please rate, review, and subscribe to our podcast on your favorite podcast platform so you never miss an episode. We'd love to hear from you, so please send us your feedback on Twitter, Facebook, LinkedIn, or via email at podcast@activemotif.com, and we'll give you a shout out in a future episode. For more great epigenetics content, check out the Active Motif blog at activemotif.com/blog. Thanks for listening and stay tuned.